Hi, I'm Vidya, and I'm going to be talking about comparative transcriptomics and hemlocks. There are 9 described species of hemlocks, 4 of which are in North America, and the others are in Asia. I'm going to be focusing on the eastern hemlock, which is a foundational species that supports unique flora and fauna. This includes the eastern redback salamander and the spotted owl. The eastern hemlock is a shade-tolerant species, and it occupies the lower canopy layers of forests. But what's killing them? The hemlock woolly adelgid was introduced in North America in the 1920s. The hemlock woolly adelgid is a sap-feeding insect that are closely related to aphids. The insect feeds on and kills all sizes and age classes of hemlock, usually within 5 to 10 years. They produce eggs on the lower half of the needles, which are covered in white woolly coating. The pulvinus or cuticle is where the hemlock woolly adelgid attacks the hemlock. Of the 9 species, the Western North American and the Chinese hemlocks in red are described as the most resistant. The Asian hemlocks show varying levels of tolerance and are denoted by the green box. The most susceptible species, the Eastern and Carolina hemlock, are denoted by the black box. For my study, I chose 4 different individuals across 6 different species. We sent needle and branch samples for GCMS targeted analysis. We also performed comparative transcriptomics. We selected targeted GCMS because high concentrations of these listed terpenoids deters the adelgid, whereas high amounts of isobornyl acetate attracts the adelgid, leading to trees becoming either tolerant or susceptible. Let's look at the results. Targeted GCMS shows a variation in terpenoid profiles across susceptible, resistant, and tolerant hemlocks. What's quite interesting to see is the huge variation of the isobornyl acetate in the susceptible hemlocks. Looking at the variation of terpenoid profiles between individuals per species, it is easy to see that the eastern hemlock and Carolina hemlock have different profiles for isobornyl acetate. One may also notice Seaboldii with a relatively high amount of isobornyl acetate production. Heterophylla also shows a variation in the amount of alpha-pinene production. What's interesting to note here is the eastern hemlock shows a high amount of isobornyl acetate production, as well as a large variation in this production. Let's look at the transcriptome analysis. Sophia is a research intern in our lab, and she generated transcriptomes for 3 different species. We ran OrthoFinder to notice that many shared orthogroups were involved in the production of mono or diterpenes and were enriched for the key enzymes in the MEP and MVA pathways, as well as terpene synthases. The unique orthogroups were involved in disease resistance. Further research is to be conducted, and we have to add 2 more species here. A big thank you to everyone mentioned here on this slide. Please follow me on Twitter and check my Git for updates.